Afternoon to honorable judges and fellow friends. I'm Gorchan Gang and I'm from Victoria Junior College, Singapore. And I'm very privileged to be here to present you one of my studies, which is titled Sulfonyl urate Agents Enhance Adipogenesis via PIPA gamma activation in human adipose-derived stem cells. Although now it can look a bit scary and very mouthful, but later on I'll elaborate more on the specific details of the title. So basically, before I elaborate more on the core of the research topics, let me give you a very brief introduction on what is diabetes. So basically, diabetes is a common disease in which the patients suffer from high blood glucose level. And there are two main types of diabetes, which is type 1 and type 2 diabetes. The type 1 diabetes is due to genetic disease, and it might be due to the damage of pancreatic beta cells, and no insulin is produced as a result. But however, for type 2 diabetes, it is different in a way that there's insulin resistance as well as low insulin secretion level. It can be due to, it is a kind of acquired disease which is highly related to obesity. But in our research studies, we are mainly focusing on type 2 diabetes. And so what is type 2 diabetes specifically? It is the major cases which are found among the diabetic patients. About 90% of diabetic patients have type 2 diabetes. Secondly, it's an acquired disease as mentioned earlier. And a high intake of rate of glucose higher than the rate of metabolism out of the obese people will eventually result in the accumulation of lipids in which lipogenesis will occur. So what is lipogenesis? Lipogenesis is basically a process in which the glucose is converted into triglycerides, which is, you call it fats. And once they are converted to fats, they are stored in adipocytes, which is adipose tissue, to give you these fat cells. So trigger lipogenesis and the storage of triglycerides among the obesity people, because they already have oversized capacity of the subcutaneous fat layers. So eventually, since it's already full, these kind of lipids will eventually be stored in the visceral fat layers, which is found between the organs, which coat the organs as a protective layer. And this is actually very damaging because the deposition of lipids on the organs will eventually cause partial organ malfunction, in which for example, liver cells and muscle cells will eventually be less sensitive to insulin action due to the deposition of lipids on them. So, and thus, it results in insulin resistance, as I mentioned, in type 2 diabetes. So, in order to treat type 2 diabetes, one of the most common drugs is thiazolidinediones, which is TZD group compound. And examples of TZD will be pioglitazone and osaglitazone. And here is the functional group of TZD compounds. So the main function of TZD is the ligands of PIPA gamma, in which PIPA gamma is a type of specific transcription factors which is very important in the process of adipogenesis, in which I will elaborate more on adipogenesis. So let's moving on to the project itself. Firstly, sulfonyl urate agents. So here I have three examples of sulfonyl urate agents, namely glibenclamide, glypyrite, and glyphosate, and they have the functional group as shown. So what are sulfonyl urate agents then? Sulfonyl urate agents are actually a type of common oral anti-diabetic drug used to treat type 2 diabetes patients. But however, they are different from TZD in a way that they bind to the KATP channel, which is the ATP-sensitive potassium channel found on many kinds of cells, including pancreatic beta cells, to actually induce the insulin secretion. So when you have more insulin present in your blood level, eventually it will trigger the storage of conversion of glucose into glycogen and stored in the liver. So binding of sulfonyl urate agents to the specific sulfonyl urate receptor on the channel will eventually stimulate insulin secretion. Previous literature actually also shows that the binding of sulfonyl urate agents onto the KATP channel will eventually result in the side effect, which is weight gain. So now we are thinking that the weight gain, is it just due to the effect of anabolic effect of insulin itself that convert glucose into triglycerides? Or is it due to another kind of effect, a more direct effect instead? And that's why we hypothesize this reaction, which is adipogenesis. So, what is adipogenesis? Basically, it's a process of cell differentiation from human adipose-derived stem cells to give rise to new adipocytes, which are fat cells. So firstly, let me give you a very simple mechanism of adipogenesis. Firstly, there will be upregulation of CBP beta and delta, in which they will bind to the promoter of CBP alpha and pipa gamma to result in the upregulation of both these transcription factors. So let me give you more insights on what is pipa gamma. Pipa gamma is basically well known as the master regulator in the process of adipogenesis. Why? Because pipa gamma regulates the expression of a wide range of genes which are required in the process of adipogenesis. And what are the examples of such genes? They are adiponectin and AP2, and these two are the pipa gamma targeted genes, although there are more. So what is pipa gamma activation then? When there's a ligand such as TZD group compounds bind to pipa gamma, not only it binds to pipa gamma, it also activates pipa gamma to result in a positive feedback between CBP alpha and pipa gamma, and both of the expression of pipa gamma and CBP alpha will eventually be upregulated. When they are upregulated, it will result in the upregulated expression of pipa gamma targeted genes, because these kind of transcription factors will upregulate the genes here. And when there's upregulation of these genes, it will result in adipogenesis, which is adipocyte differentiation. Next. Yeah, this is human adipose-derived stem cells, in which they are derived from the adipose layer, which is at the subcutaneous fat layers. And they are multipotent mesenchymal stem cells, in which they are able to differentiate to give rise to a limited range of specialized cells, such as adipocytes, chondrocytes, and osteoblasts. They are a range of cells which are present in a particular metabolic pathway. So what are the objectives then? Firstly, we want to investigate whether the sulfonylurea agents, in which in my case, they are glibenclamide, glypyrite, and glycoside, whether they are able to enhance adipogenesis similarly as TZD group compounds. Secondly, we also want to determine whether these three sulfonylurea agents could actually bind to pipa gamma to result in enhanced adipogenesis as a result, which is also similar to TZD. Thirdly, after doing all this and we find out that they actually enhance adipogenesis, they actually bind to pipa gamma. Now we want to see whether is there any increase in mRNA level of the pipa gamma targeted genes after they bind to. Because we not only want to know the binding activity, we also want to know the efficacy, in which whether the activation extent is there or not. So for the research methodology, the main bulk comes from the cell culturing. After the cell culturing, we actually do this oil red O staining together with crystal violet staining. What is the purpose of oil O staining? It's basically to stain the adipocytes form differentiated from the HASC, so we can measure the absorbance value after extraction so as to determine the amount of adipocytes formed in the wells. So by doing this, the absorbance value will actually be proportional to the amount of adipocytes formed, and hence the rate of adipogenesis. So secondly, we also do the pipa gamma ligand binding assay so as to determine the binding activity of the three sulfonylurea agents, as well as the positive control, which is pioglitazone, a type of TZE in my experiment, so to see whether they actually bind to pipa gamma. And after that, we do qPCR to determine the mRNA level of the two pipa gamma targeted genes. And for three types of sulfonylurea agents, we actually use three different concentrations, which is 0.1, 1, and 10 micromolar. So for the adipogenesis results, which is oil O staining, as you can see clearly from this graph, this is the absorbance value, and this is the concentration of the glibenclamide. So you can see very clearly that with increased concentration from 0.1 to 10, there's an increase in the absorbance value, which means there's an increase in the amount of adipocytes formed, and hence adipogenesis. So you can see, if we compare to the positive control, which is pioglitazone, it's quite comparable level of adipocytes formed. And you can see very clearly from these pictures, sorry for the cut off, but this one is the positive control, and this one is the glibenclamide at 10 micromolar. So the red dots and red patches actually represent the adipocyte stain with urate ore. Similarly, for glypyrite, they actually show a very similar trend to glibenclamide as well. And there's an increase in adipogenesis, enhanced adipogenesis, when there's an increase in concentration. However, for glycoside, we can see that the increase in the absorbance value is not very significant in this case. So actually, glycoside might not be a very suitable ligand to pipa gamma to enhance adipogenesis. So after looking through the adipogenesis results, we want to look through the specific details and the steps within the reaction itself. So for binding activity with pipa gamma, you can see very clearly the positive control pioglitazone has a drastic increase in the binding activity with pipa gamma with increasing concentration as compared to the 3 other sulfonylurea agents. But having said that, we should not ignore the fact that there is actually some increase in the binding activity. So you can see from these 3 sulfonylurea agents, the blue one is the glibenclamide, red one is the glypyrite, and this green one is the glycoside. So you can see that for glibenclamide and glypyrite, there's a significant increase in the binding activity to pipa gamma when the concentration increases. So we can be sure that these sulfonylurea agents, they are indeed, agonists or ligands to the pipa gamma. So for qPCR results, we can see also, for glibenclamide and glypyrite, they exhibit increased mRNA level of AP2, which is likely comparable to pioglitazone as well. What about adiponectin? Yeah, we also obtain positive results in a way that glibenclamide, they show an increase of adiponectin mRNA, similarly as GMP, as compared to the MDI, in which there's no treatment. So basically, you can say that glibenclamide and glypyrite, they not only bind to pipa gamma. After binding to pipa gamma, they actually activate pipa gamma to result in the increased expression of these 2 types of pipa gamma target genes. So this is a piece of extra information. We did this reverse transcriptase PCR on the undifferentiated HASC, and we found that, as I mentioned just now, this KATP channel is a large octameric channel made up of 2 types of subunits, which are SQR subunits and KIR 6.X subunits. And these 2 types of subunits, they are actually present in different isoforms, in which we detected SQR2B and KIR 6.1 in HASC, which is this. Although the map is very, very faint, I admit, but it's actually observable, just that maybe the quality of the projector is not there. But although we see on paper, it is quite faint as well, but it is a type of preliminary results in a way, that this is the first results ever published, and no one has ever discovered whether KATP channel is present on HASC or not. So we can actually say that because there's this combination of SQR subunits and KIR 6.1 subunits, we can say that actually KATP channel is indeed present on HASC with the undifferentiated ones. So to conclude, we know that from the results itself, sulfonylurea agents, they bind to pipa gamma, they possess binding activity to pipa gamma, as observed from this graph. Secondly, glibenclamide and glypyrite, they also upregulate the expression of pipa gamma target genes, which are AP2 and adiponectin. Thirdly, without a doubt, we can say that glibenclamide and glypyrite, they actually enhance adipogenesis to a significant extent. Lastly, we also detected that perhaps there might KATP channel might be present on HASC. So, what does all these results actually give rise to? Actually, it's raised up a very interesting point that how come pioglitazone, which such drastic increase in the binding activity, will still result in a comparable adipogenesis effect? How come? And this raises up a question. So we actually hypothesize that is it possible that the adipogenesis effect triggered by sulfonylurea agents, they're actually mediated by KATP channel? Because we already know from previous literature that sulfonylurea agents, they do bind to KATP channel. So there is a possibility, but we still do not know what is happening in this black box here. So with this renewed understanding about HASC as well as the sulfonylurea agents, there is a need for us to rectify whether KATP channel is really present in the reaction of adipogenesis, whether they are participate in the metabolic pathway. And even if it is involved in the particular pathway, we also want to know what role does this KATP channel play in adipogenesis. Secondly, we also want to discover the potential of sulfonylurea agents so that we can actually synthesize or design a novel anti-diabetic drug. Because previously we know that sulfonylurea agents, they only stimulate insulin secretion level. But now, from this result, it actually acts similarly as the TZD group compounds. They actually improve insulin sensitivity in a way. So can we actually create a novel anti-diabetic drug that can actually increase insulin secretion as well as to improve the insulin sensitivity as well? And I've come to the end of my presentation. Thank you for your undivided attention.
Any questions from the floor? Judges?
Thank you for a very illuminating sharing there. Just out of curiosity, would like to know, at the end of your research, would it lead to the development of a new drug to assist in combating those with type 2 diabetes? Just as a matter of curiosity.
There is a potential there, but there's a lot of future work need to be done before we can reach that stage. Because this is still at the research stage, eventually needs to go to the application stage, which will make more viable. Because we still need to consider the concentration of the sulfonylurea use. Because we need to consider the half-life that's present in the body, how long can it last in the body. So we need to consider all these cases in order to actually come up with a new anti-diabetic drug in the future, which can be prescribed to type 2 diabetes patients. Because we need to also take into account the fact that these kind of sulfonylurea agents, it can result in weight gain. Because we already know that diabetic patients, they are obese in a way already, right? So if you give them sulfonylurea, wouldn't they become fatter or something? That is not very desirable. So I think, although there is a case in Japan, because this research is mostly focused on Japan. Because Japan, they have population which have a lot of people having diabetes type 2. But they remain very thin, they are not obese, but they have diabetes type 2. So this might be a very possible drug in the future for them. Yeah, I hope I answered your question.
Thank you.
Thank you very much.